Ladies and gentlemen, welcome this afternoon. On behalf of Invivoscribe, we welcome you to this portion of AMP Global 2019 Special Symposium. We appreciate your time and taking the effort in being with us and joining with us today. We hope that you'll have an enjoyable time and educational time as you participate in this symposium today, and we encourage you at the end of the speakers to ask any questions that you might have. Today's session, NGS Solutions for the Assessment of Clonality and Somatic Hypermutation in Hematologic Malignancies. We're very pleased to have two special speakers joining us today, both of them coincidentally from Germany, so they came a long distance. The first speaker, Dr. Bettina Budeus, from the Group Molecular Genetics, Essen University Hospital in Essen, Germany. She will be speaking to us about the analysis of human BCR rearrangement repertoires in health and disease. Our second presenter, Dr. Roshana Kabab, is coming to us from the Berlin Reference Center in Berlin, Germany. And she'll be talking to us about practical interpretations of clonality results using identiclone and lymphotract assays in daily diagnosis. Again, we encourage you to pay attention and take notes and also be able to ask questions if you should have some at the end of the presentation. And with that, I'll turn the, I want to make one other announcement. You might also have noticed on your programs that tomorrow afternoon we have an additional session at 12 o'clock noon regarding MRD disease and somatic hypermutation. We hope you'll also be able to find time in your agenda to attend that. So with no further introduction, I'll turn the time over to Dr. Bettina Budeus to begin the presentation. Thank you.
Hello, everyone. Hepatitis C virus does not only induce liver disease, it also can lead to autoimmune diseases like mixed cryoglobulinemia type 2 or B-cell non-Hodgkin lymphomas like diffuse large B-cell lymphomas, splenic marginal zone lymphomas, Waldenström's microglobulinemia, and follicular lymphomas. The aim of our study was to analyze the alterations of the B-cell subset compositions, the biases in the BCR receptor repertoire of the main B-cell subsets, and to analyze the presence of clonal expansions. For this, we took HCV positive patients without MC and analyzed the B-cell subsets, naive, class-switched memory, non-class-switched memory, and CD5 mature cells for their clonal composition. So here's our study flowchart. We had 30 HCV positive patients and 15 healthy donors. We extracted the CD19 positive B cells via MAX, then used the flow cytometry to analyze the B cell subset frequencies, and did a cell sorting only for 22 HCV patients and 7 healthy donors because they had sufficient cells to use high throughput sequencing of the AGHV gene repertoire. This is our baseline characteristics of the HCV infected patients. So the median age of our patients was 33, and the gender distributions was more or less equal with 14 to 16. Our HCV genotype was mainly genotype 1 and 3, and only 3% had genotype 2. And as you can see, we also searched for cryoglobulins, but none of our patients had any. This is our flow cytometry and cell sorting strategy. At first, we used the live gate for our cells, then we ruled out doublets. And to be really sure that we have only B cells in our analysis, we used CD20 positive B cells only. Then we subdivided in CD23 and CD27 expression. So those negative in both are CD27 negative memories. The CD27 positive ones are subdivided by IgM expression into class-switched memory and non-class-switched memory. And the CD23 high expression ones are subdivided by CD5 and CD38 into naive, CD5 mature, and transitional B cells. Here you can see the analysis of our subset distributions. So they are more or less the same between HCV patients and healthy donors, except for 3 subsets. So we have an increased frequency of class switch memory B cells in HCV patients and a decreased frequency of naive patients and CD5 transitional B cells. Then we did our clonal analysis. So for this we used only 22 HCV positive patients and 7 healthy donors. Used cell sorting to extract naive class switch memory, non-class switch memory and CD5 mature B cells. Then extracted DNA via QIAGEN kit, subdivided them into 2 replicates with each of about 10,000 B cells. And then to generate the library for the sequencing, we used the Invivoscribe lymphotrack framework region 1 assay. And then sequenced it in an Illumina MiSeq. So this kit uses for framework region 1 primers for all families, so VH family 1 to 7, and reverse primers for GH family 1 to 6. So with this we are able to analyze mostly the whole V gene for somatic hypermutations, the complete CDR3 region for the whole, the length distribution, and of course the clonality analysis. You have mostly 3 kinds of clones in your sequences. So there are clones with interclonal diversity, arranging from the same subset, but with mostly the same sequence, but minor mutations inside the sequences. Then you have clones without interclonal diversity, which are also from the same subset, but without those mutations and are thus hard to find. And you have polyclonal cells, which are from different subsets. So all of this you have in your sequences. You generate DNA and the library from this and get amplicons, and then sequences and have the problem to find the clones inside. So we have written our own bioinformatics software for this, which does a VDJ search for this. So it identifies the V gene for the sequence, the J gene for the sequence, and then searches for the CDR3 inside. And all those sequences with the same V gene and the same J gene and an identical CDR3 length and identity of 90% in CDR3 is called a clone if we find it in both replicates. This is easy if you have little mutations inside, but if you have clones with intraclonal diversity, without interclonal diversity, then this is hard to find because you want to collapse identical sequences which can be the result of PCR amplifications or sequencing artifacts. So you collapse them normally, and then you would only get one sequence, and one sequence is no sequence actually. So we use sophisticated bioinformatics and statistics to get a threshold above which we consider a clone without interclonal diversity as a real clone if we find reads in replicate 1 and replicate 2. Here you can see some numbers for our reads in the patients. This is patient 30. So we have 10,000 cells in each subset, and we got between 600,000 and 800,000 reads from these cells. Then after quality control, we had between 400,000 and 600,000 reads. And from those, in this case, we find between 3,000 and 6,000 different clones. So we also checked how many functional rearrangements we have here, and this was in this case between 4,000 and 6,000. In other cases, we had between 2,000 and 19,000 productive rearrangements. The first, what we did analyze was the VG gene usage, and this is mostly identical between the subsets, except for 3 classes and families. So the first one, BH169, was increased in non-class switch memories. The second one, 330, is increased in non-class switch memory, CD5 positive and naive B cells. And the third one, BH459, is also increased in non-class switch memory B cells. And they are all said to have an influence in autoimmune diseases. So this was a really interesting finding. Second, we just analyzed the somatic hypermutation frequency, which was, as expected, higher in the memory B cells than the naive ones. But we found that the naive B cells from the HCV patients had a higher mutation frequency than those from the healthy donors, which could, of course, be an artifact from just memory B cells sorted into this, but it could also mean that there's something going wrong in the immune system in those patients. And third, we analyzed the CDR3 length distribution, which is again, as expected, higher in the naive ones than the memory ones. But here you can see that in the class switch and non-class switch memory B cells, the CDR3 length distribution, they are longer in the patients than in the healthy donors. And we think that this indicates that there's something going wrong in their germinal centers. So they are not fully working anymore. And you can see what we also analyzed is that there's a positive correlation between viral load and CDR3 length. So the more virus the patient had, the longer their CDR3 length was. Then we analyzed the clonal composition. And here you can see on the x-axis the member count in clone, and on the y-axis the read count in clone. So this is the size of the clone and how many reads we had in there. Also, as expected for the naive and CD5 positive ones, many small clones with low numbers, and for the memory ones, larger clones with more numbers. But especially in this case, for the non-class switch memory, you can see there's a huge clones of high sizes with a lot of reads in the HCV patients. And here you can see it in another representation where, especially for the class switch memories, you have lots of percentage of reads of expanded clones compared to the healthy donors. Here you can see we also did some tree representations with IG tree. The root is always the baseline, the germline sequence. Then you have small dots here indicating the mutations, but we didn't find those sequences. And as squares, you have those sequences we found in white in replicate 1, in black in replicate 2, and those in gray are found in both replicates. This one, this really large one, it's above 50 sequences, was the second largest clone we had in the HCV patient, and it's from non-class switch memory B cells. So just to give you some numbers, those are the individuals with clonal expansions. Here clonal expansion means 5 sequences in each replicate, so a total of minimum 10 sequences. And for the naive and the CD5 positive ones, we only found them in the HCV patients. And in the non-class switch and class switch memories, we found far more patients with such clones, so 17 compared to 6 and 13 compared to 1 in the HCV patients. Also, the average number of clones per individual, you have in the non-class switch memory 6 clones, and in the HCV patients and in the healthy donors you have only 3.6. And for the class switch memory it's 2 versus 0.1. And the number of large clones, so clones with more than 30 members, is 14 in HCV patients and only 1 in healthy donors. So to sum it up, chronic HCV infection causes extensive BCR gene repertoire alterations with pathological features even in the absence of mixed cryoglobulinemia. And many large B cell clones are consistently found mainly among IgM positive memory B cells, showing a massive influence of HCV on this compartment. And with this, I want to thank especially Dr. Tucci for doing the sample analysis, sorting, and all the lab staff, and Daniel Hoffmann and Simo Kitanowski for analyzing the sequences we had there. So if there are any questions, I'm happy to answer.
How do you design the primers for the amplification?
No.
And also, how many pairs of primers did you use?
No, the primers are inside the lymphotract kit, so we just used it as provided.
So they provided your primer? Yeah, actually, that's a question maybe for the vendor. You know, do they have people here? Yes. Yeah. So how do you design? What's the first? How do you design primers? How many pairs primers in using study?
Well, the number of primers as proprietary information, you know the Biomed2 primers, Biomed2 technology, right? So we amplify the same regions, but the sequences and the number of primers, that would be proprietary information.
That's fine, but how big
How big? Oh, she's testing framework 1.
Yep.
Only. So the amplicon size will be about around 300 bases.
Yeah.
Yes.
Do you look at the immune repertoire after treatment of the hepatitis C patient?
No, there are only non-treated patients we analyze here.